everyone. I am Saniya Khullar, and this is cute little Yangshi. And in this video, we're going to be learning about a very cool tool that will be able to help us understand better what is the impact of a SNP, a single nucleotide polymorphism. So how can we map human SNPs using rsIDs, which are basically like another way, begin with RS, and it's sort of corresponds to a chromosome number and a position. How can we use those rsIDs and then link it to annotated DNA regions and gene names, and then predict their impact? So we're going to be using this really cool tool that is available in gProfiler. And we're going to be doing this. So this is kind of figuring out basically what is the impact of a SNP. Okay, so basically, just to explain a little bit, we have DNA regions. For instance, we have like regulatory sequences like enhancers, silencers, promoters, 5' untranslated regions. So when we are reading DNA, there are multiple factors like transcription factors and RNA polymerase. They're helping us transcribe DNA into messenger RNA that then leaves the nucleus and can be used in various ribosomes as well to come up with proteins, and they have various functions. So basically, if you look at DNA, if you look at the central dogma of molecular biology, we can look at DNA being transcribed into messenger RNA, and then that messenger RNA has some sort of modifications that are made, and then it is transcribed, after it's being transcribed here in the process of transcription and then mRNA processing, messenger RNA, it's then translated into proteins or polypeptide chain by ribosomes here. So we can see that there's like a huge process involved in many regions along the DNA, enhancers and silencers, promoters, 5' untranslated region, open reading frame, intron, exons. The introns are removed and the exons are spliced together. And there's also some alternative splicing, and I can make more videos on that too. But the exons are kind of put together in the 5' cap and the poly A tail, and then they can help form this mature messenger RNA that is then translated by the ribosome into like a polypeptide chain or a protein. And so if we look at the DNA regions here, so if we look at what is in our genome, there's so much. There's centromeres, pseudogenes, introns and non-coding RNA genes, possible non-coding RNA. There is so much, and protein coding genes are so small, like 1%, unknown, defective virus sequences, functional transposons. There's just so much that's going on, the pie chart of what is composing our genome. So essentially, this tool that I want to show you is gProfiler, and it is bit.cs.ut.ee/gProfiler/SNPense. It helps go from SNP IDs that are given in the form of rsIDs to an associated gene name. So it maps a list of human SNP RS codes like RS7961894 to gene names. So it gets the chromosomal coordinates and the chromosome number and the base pair, and it helps predict the variant effects. So SNPs are single nucleotide polymorphism, the most common type of genetic variation. So, it's a single nucleotide difference in a particular spot in the genome here. That's a SNP. A single nucleotide polymorphism. So this is a SNP. Is in one person it can be an A, another person it can be a G. So this will show you how we can do that. So basically now I just put a little warm wet cloth on Yangshi so I can help him feel a little bit better as well while he's learning as well. Apparently it reminds these little cats of their mothers as well. So it's nice and warm for him. But basically, what I want to talk about is SNPense, which is helping us to make sense of SNPs. SNPs are single nucleotide polymorphisms or a single nucleotide variant, SNP. So you can see here, so we're trying to make sense of SNPs, and the tool is SNPense. So If you look across the DNA, there are these 4 main nucleotides. Adenine pairs with thymine, or A and T. Guanine pairs with cytosine, G and C. So the sugar phosphate backbone here. And what a SNP is one of the most common types of genetic variation. It's like a mutation, a single nucleotide is different. So it can be a C here in one person and a T here in another person here. You can see like these examples of SNPs. So if we look at 2 people here and we look at their DNA sequences, what we can see is in the SNP situation that there's like, you know, in this person here, this is an example of 4 SNPs here. 4 SNPs, 1, 2, 3, and 4. So it's an A in this person blue, but a G in person green. It's a C in person blue, but an A in person G, and a T here, but an A there, and a C here, and a T there. And the rest of the sequences seem to be similar. These are just one nucleotide difference. And there are many different types of variants as well. But what is the impact that they have? What is that? If one person has an A and the other person has a G, what is the impact of this mutation? And what we see is in the human genome, the DNA, so much of it, these are protein coding genes, but so much of it is non-coding DNA. The introns, there's miscellaneous unique sequences, heterochromatin, segmental duplication, sequence repeats, transposons, retrotransposons. There's just so much. So what is the impact of a one DNA base pair difference, and what genes is it linked with? So we're going to be using this tool called Snippens. It's by gProfiler, and it's one of many tools that are out there to help us, link to genes. It helps give us also from SNPids to HG38 coordinate positions as well. So it's just one of those components. And it's really important as well because we can better understand the impact of the SNP. Okay, you on, buddy? He's feeling a little bit better. So basically, we can look for mapping as enabled for variants that overlap with at least one protein coding ensemble gene. And they're using the ensemble variation data here to do this. So basically, they're looking at the effect of the SNPs, looking at sequence ontology ordered by severity. So it can be transcript ablation, splice acceptor variant, splice donor variant. So it's kind of showing you that there's so many DNA regions. So the impact of the SNPs can be, so yeah, there's so many DNA regions here, so much that is going on, just so much complexity as well in the DNA. So of course, the SNPs, like the mutation, there are a change, one DNA base pair difference in a particular chromosome and location, single nucleotide polymorphism, one nucleotide being, like one nucleotide being different in a particular chromosome and base pair position. So it's quite common type of variation. So these could fall like in splice acceptor variants, frame shift variants, stop loss, start loss, transcript amplification, in frame insertion, deletion, missense variant, protein altering variant. Maybe this is not heard of. Splice incomplete terminal codon, 5' UTR variant. So we're trying to figure out where are these found. So you just have to input a list of SNPids here. So I'm going to show you an example where we can run their example and then predict what does this list, a list of SNPs. So SNP given these rsIDs here, this is just example query. We can try to see what do these list of SNPs do. Maybe this list of SNPs is significant from a genome-wide association study, for example. And then what we want to do is just figure out like what do they tend to do. So I was going to try to map them. So you can see that this first one in the list here, this RS 117343132, nothing was mapped to it. Nothing really key. We don't really know what it is all about yet. And that can be a key thing maybe you look into. Does this SNP have any known impact? Because a lot of these are using databases and what we found. So this other SNP here in chromosome 12 in this position here. So SNPs are just one. So this other SNP here, this SNP is corresponding to chromosome 12, start and end. The the width of a SNP is just 1. So the start is always going to equal the end. These are always going to be equal because a SNP is just one nucleotide variant. So along a chromosome at a particular position, so start and end are just the same here for the SNPs. So that's what we look at over here. And what we can do is we can try to figure out what is the impact of the single nucleotide polymorphism. What are the impact here? So this is in chromosome 12 in this position 1, 2, 1, 9, 2, 7, 6, 7, 7. So just to kind of show you guys, if you look along chromosome 12, it has about 132 million base pairs. And we try to figure out, okay, well, what regions, Alzheimer's disease, taste receptors, all of that, 132 million base pairs here. So if we look along in this region here that already has 132 million base pairs, if we try to see for this SNP, what it is associated with, and this position 1, 2, 1, 9, 2, 7, 6, 7, 7, this SNP is associated with this gene name at this position 1, 2, 1, 9. It's associated with this ensemble gene ID, which is the same, which is another ID. There's Entre ID, there's ensemble gene ID. So we can say that it's associated with this gene. And if we look up this gene, indeed, it has many sorts of IDs. It has an HGNC, it has an NCBI Entre gene ID. I'm a huge fan of these. It even has this ensemble ID, which we see here. It ends in this ID here, which we see will be the same thing as what this is here. So what we're going to then see is it's mapped to this gene. And it's an intron variant for this gene CFAP251. And it's also a non-coding transcript variant. So that's what we sort of see. We see the SNP, we see the gene. So from the SNP, we can also get chromosome number, start and end position information as well. So that's one thing. If you're given a list of SNPs, you can then find the chromosome number, start and end position. And I have other videos on how you could also potentially do that too. Chromosome number, start and end positions. Then we also have the gene IDs that they're associated with, which also correspond to the gene name. So you can use either or. And we also figure out what they are associated with that gene. If they're intron variants, if they're non-coding transcript variants here, for instance, for CFAP251. So then another thing we can also see, so that's what CFAP251, that's what this SNP is associated with this gene, particularly in this region, intron variant and this. Then this next SNP, RS4305276, is associated with chromosome 2. And with this position here, you see that they're the same, 240559596, and it's associated with this gene. Based on the coordinates that we know, it's found in an intron variant again, and with a non-coding transcript variant. So we can see this information here. We can also export to a CSV, but we can see that this SNP, nothing was found for it. So the ones that are light gray, nothing was found. But for the other SNPs, this one here, we can see, okay, this SNP maps to exos C3L4, and it's with an NMD transcript variant here. It's with an intron variant here, and it's a missense. No, not a missense, but it is also a non-coding transcript variant. Then this one here is associated with an ensemble gene. So we'll have to figure out what this does. This SNP is associated with multiple genes here. So if we download the CSV, we can see this more clearly, but it's linked with SH2B3 and ATX2, and it's associated with NMD transcript variant, with an intron variant, with the missense variant. And then this one's missing, this one's missing. This one is located in chromosome 2 at this position here, and it's linked to GCKR, and with the missense variant, with a non-coding transcript exon variant here, with a non-coding, so it's linked to missense variant, non-coding transcript exon variant, and then the last category, splice region variant. So there's so many, these are the main categories that are being considered here. So B-farm UTR, NMD transcript variant, intron variant, missense variant, non-coding transcript exon variant, non-coding transcript variant, splice region variant. So there are all these SNPs that you can map. and you can see what these are doing. And these are the variant effects that describe what color-coded set of variant consequences terms here, as I was mentioning. So this way you can figure out SNP, the chromosome number and position. You can also figure out the gene IDs that's associated with the gene names. And it takes as input rsIDs here. It needs rsIDs. So we can export this to CSV. And if we export it to a CSV, we can then open it up here really neatly. And we'll get the list of information, the strand information. If there's nothing found, it'll just be -1, -1. And we can get the severity as well for these. But this is a way to figure out what your SNP does. Like where does it get mapped? So if there's no information here, we can just remove that. But for ones where there is information, we can figure out, okay, this is an intron variant here. This is a non-coding transcript variant. We can figure out which are the associations between the SNPs and genes, in particular, which regions do they impact. So this is a really, really cool tool that I found that I thought would be helpful because we can better understand the impact of a single nucleotide polymorphism in these regions here. So hopefully our research can help build it out as well. And they're color coding these values here, they are belonging to this color coding scheme here. Transcription factor, ablation, transcription factor, binding site variant. So we can try to understand their impact this way as well. And we can also export to PNG. And that way, we can also just get this whole thing down. We can get this entire image instead of taking screenshots, we can get it this way. So these are some of the effects that we can sort of predict here. And it's using data from Ensemble as well. So if we download the CSV, you can also see the situations where the SNP is mapped to multiple genes here, which is also really key. And you can use all these effects over here. So essentially what this is making sense. This is mapping a SNP to what it is involved with. This is mapping a SNP to what it's involved with. It's finding this information and building SNP RS identifiers to genes. It's really cool. And what you can do is you can also run this in R as well. You can have a bunch of SNP IDs that you want to translate and find what they are associated with. So this is just Snippens. So it's making sense of what these SNPs are, going from RS identifiers to genes and finding out their effects on variants. This is a really powerful tool here that you can figure out all of this information from. And you'll see here, if you use the Snippens in R using Bioconductor, you just have to put in the list of the SNPs that you're interested in. And then you'll be able to run the result and you'll have your rsIDs. If it's not mapped to anything, it'll just not show anything here. Even though a SNP maps to a chromosome in a start and end position, it just won't show anything. You can just skip it. It's only focusing on the SNPs that was able to successfully map here. And then you can just see, the associated gene names, an NMD transcript variant, variant introns variant, variants missense variant, variants non-coding transcript variant. So the ones where it's 0, you can ignore them, but whenever there's some number, you can use the number. So it's a data frame with corresponding variant effects. So the reason for this is that biological data analysis often deals with lists of genes arising from various studies. The gProfiler toolset is widely used for finding biological categories enriched in gene lists, conversions between gene identifiers, and mappings to their orthologs. So there is a lot of information here. And if we look up, Snippens is going to map human SNP identifiers to genes. So it allows the user to easily map a list of human SNP RS codes like this, for instance, the gene names. We can get chromosomal coordinates, predicted variant effects. So mapping is enabled for such variants that overlap with at least one protein coding ensemble gene. We only focus on the protein coding genes. All underlying data are gotten from ensemble variation data. The variant effects are described with color-coded set of variant sequence consequences terms that is defined by the sequence ontology. These terms convey the information about the effects that each allele of the variant may have on each gene. So if we check out the sequence ontology here, the sequence ontology is 29 here. Then we can see here improving the sequence ontology terminology for genomic variant annotation. So you can see that these are the sequence ontology. It's very complicated here. The sequence variant, structural variant, feature ablation, feature amplification. There's just so much TF binding site. There's such a hierarchy. There's such a hierarchy of what's going on here. And we can get definitions for these terms here. Missense variant. So what a missense variant means is it's basically where codon bases are modified in such a way as the resulting amino acid would change. And splice donor variant. So a missense is like a mutation where the effect of the SNP, the mutation can change the amino acid that the ribosome will read because the messenger RNA is affected. Splice donor variant alteration changes a 2 base pair region at the 5' end of an intron with the splicing that is going on. There's so many different terms. There's a sequence variant node, structural variant, and functional variant. So if we look in the table, there's structural variant here under the sequence variant and functional variant. And structural variants could be changes with regard to annotated sequence features. So there's just so much that is going on over here. So you just need the rsIDs, and then you could figure out what those are indeed involved with. So basically, this is cute little Yangshi, and this is me. And we just hope that this has been helpful for you to figure out how you can go from a list of SNPs to figuring out their chromosome number and position and what their effect is on those genes that they're mapping to. What regions could they be annotated to? So how can you link SNPs to that information, the SNPs to the variant effects? And there's so many things. SNPs are single nucleotide variants, or also known as single nucleotide polymorphisms. So how can we predict their effect and link them to genes and also figure out what they are involved with? So this is just a really cool tool as well. So I just hope that this was helpful. There's so much else. I also have another video where I'm talking about how we could do functional profiling for genes. So this one for Snippens, you can make sense of those SNPs. So for all these SNPs around, you can make sense of what they're doing. If you see a list of these SNPs, see, yeah, she's pointing to it above, a list of SNPs, you can figure a single nucleotide, a list of SNPs. So if you have a list of SNPs, so Yang-Chi's pointing to them here. You have a list of SNPs, you can single nucleotide polymorphism here. You can sort of figure out what those are involved with. You could figure out what those are involved with and what are the regions, the genomic regions that I have listed here, like right here, like what regions are they involved with? So this is a very helpful tool. So gProfiler has a lot of cool things that are going on with it. So I hope this was helpful. And if this was helpful, please like and subscribe to my channel. And please let me know if you have any questions at all. So there's just so much that you can do with gProfiler. So I hope that it's helpful and that you can use these tools to better understand the effect of single nucleotide polymorphisms in the DNA regions along all the chromosomes, what genes are linked with, what is the impact that we know about. And this is ongoing research. People are trying to figure out what these are involved with. So please do, try this. Let me know if you have any questions. And there's so many tools I want to help you guys with. And there also is the R version, and I'll try to put up a video on that too, because that involves some programming. But it might be a little bit for those of you who have questions with Python, I'll try to see other tools, but R is very phenomenal in the type of packages that it has. So I hope that this is helpful. So thank you guys for watching. And Yangshi also hopes it was helpful. Oh, so Yangshi also hopes that this was helpful to you guys. Thank you guys so much for watching. And I hope it helps you guys better uncover what your list of SNPs are doing, whether from a genome-wide association study or just some statistical analysis you were doing. I just hope it helps you figure out in a particular chromosome and a position, what do we know about that? So using other tools that I've talked about, you can go from that to SNP IDs, and then you can use this. Aww. Thank you guys. Also, it is really important to note the basically what is the genome build of the coordinates that SNPense returns to us. So I have another video where I talk about that and how you can do liftover analysis as well, and how you can tell this. So I just want to quickly show you. So please go check out. So what we can do is we can sort of let's paste this here. And let's get the corresponding coordinates as well for this SNP that we were given here. So it's chromosome 12 position. This is the position here. So we're just going to write it as CHR12, comma, this is the base pair position. So let's try HG19. And I have other videos, so please see my channel. I'm trying to put as much content to help you as possible. So this liftover analysis that can help you go between kind of like translating the coordinate system from HG19 or GRCH37 to a newer HG38 or GRCH38 version. You can do this using liftover analysis. You can do this conversion. But it's important to know the genomic coordinate that you're using just if you need it for some downstream analysis. People might be using different builds. This is something that I also learned the hard way. So I put up a lot of videos to try to help you guys with this as much as possible. So please do see my channel for liftover analysis as well. But yeah, you can just basically, I have another video as well to figure out the build using the SNP IDs. But you can just see that this is, you can put in these IDs here and you can see if it's found in HG19. So what we see here is that the SNP ID here, which is the same thing here, what is the position that it maps to? So what it maps to, it wanted it to be 1219, 27677, but instead it maps to this, which is not the same. It's not the same. Upset about that. We're upset. It is not the same. So it's not the same. And what is moreover is that it's saying, hey, if you look up 1219276777, it's saying it's not found in HG19, but it is found in HG38. So that can clue us in that, hey, this is actually found in HG38. So let's use that instead. So what we can also do is similarly, we can just verify that that makes sense. And we can do this for as many number of SNPs as you need to just feel better about it. Like, so HG38 instead, let's try that. So we're going to submit this. And indeed, what we find is it all makes sense. This same SNP here, this SNP, and this SNP here, they map together to the same position here, 121977 is exactly what this, for the same chromosome, it's just a matter of which position you use along it, and the genomic coordinates can differ. So what this tells us, guys, is that when we're using that SNPense, or SNPsense, that this SNPsense thing is sort of returning results for us in HG38 coordinate. The chromosome numbers will always be the same. It's just a matter of the coordinate system. And I have more detailed videos on that, positions in HG38, which is GRSCH38. So you might be wondering, okay, well, why is this? Or if I'm giving you some needless information, but you never know when you need to know this for some analysis. Sometimes SNPs in GWAS studies are typically in HG19 or GRCH37. That's just the older format. Sometimes some data sets like expression quantitative trait loci could be in either. So these are just some things that I've noticed generally, but you also just want to be really careful that you know this in case you're trying to do some analysis with these intervals and you have data, for instance, that if you're using these intervals, but instead your data is HG19, then maybe things don't line up. Your other data is that the coordinate systems could be different. And then you need something that is called liftover analysis. And I have a video on that, so please see my channel for liftover analysis.